Okay, so going back to company presentation, I'm glad to welcome Dorte X, the CEO of Pila Pharma. Welcome.
Thank you very much. I'm happy to be back in Stockholm today on a sunny day. Really great. So today I'm here to present our first project, a diabetes project, and nothing could fit better than be part of your diabetes event today. So thanks for the invitation. So let me see here. So just a little brief introduction to us. We are a Malmö-based company. We are clinical stage. I founded the company back in 2014, and we also have a wholly owned Danish subsidiary. We listed on Nasdaq First North last summer, and hashtag Pila for those of you who want to check us out. And we have one clinical lead candidate called XEN-D0501 that we in-licensed in 2016. Last summer we got 2 years ago, we got a proof of principle of this molecule in diabetes, showing that it effectively could improve insulin secretion in type 2 diabetics. And to add to a pipeline, so to speak, or we consider this molecule maybe a good platform to extend into other projects. We sought and got an orphan drug designation for the molecule in another indication, named erythromelalgia, a rare disease with pain. I won't talk so much about that today, but why diabetes? Well, today we have heard more about that, but type 2 diabetes is 90% of all diabetics in the world and 10% is type 1. We primarily focus on type 2 diabetes. We think we may have a possibility also to do something good in type 1, but that has not been in our focus right now, but we are beginning to look into that. But the reason for looking into these new treatments for diabetes, it's not because they're not good treatments already, but there is a growing number of patients around the globe who have a need for better access and more affordable treatment. And what I discovered 20 years back when I was a scientist at Novo Nordisk, I found out that there was potentially a new mechanism of action in diabetes where you could regulate underlying inflammation and thereby regulate diabetes, but also some of the comorbidities. And I think still that this could be a maybe not superior, but at least very good additional treatment option. And that's why I took this into my own hands, and we're now developing it ourselves. We did 2 clinical trials until now, and this is the slide that I've shown sometimes before. It's a slide from after 1 month's treatment with our molecule. We did an oral glucose tolerance test in the end, and we saw that the patients who received the dose of 4 milligrams bi-daily for a month that had an improved insulin secretion with statistical significance. What we also saw in the same study was that these patients were very obese and very insulin resistant, that we actually also got a tendency, actually this was significant for a reduction in the 2-hour blood glucose levels, suggesting that we may also have an effect on glucose. This was a 1-month study, but we did test and measure HbA1c, which is the measure for long-term glucose regulation. And as you can see from before to after treatment, there is a trend for reduction after our treatment. This did not come out statistically significant. We need a 3-month study to do that. And that is the next step for us. So the molecule we work on is it's a small molecule, and it's formulated right now in some very small tablets. And here you see a bottle from our first trial. And we have just recently completed a 3-month toxicology package where we have shown that it was indeed had a very, very good safety also after 3 months in 2 animal species. So we are now ready to move on to a 3-month phase 2b study in diabetes. The molecule has been in 8 clinical trials and actually in 300 patients altogether. And it has been demonstrated a good safety profile until now and signs of efficacy. XEN-D0501 is a TRPV1 antagonist, TRPV1 antagonist, and TRPV1 is the molecule that can sense hot burning pain. It's also called the chili receptor, and This molecule was developed by Bayer, and we took it over in 2016. And this slide just I picked this illustration from the Nobel Prize homepage, and as some of you may recall, in December 21, two researchers shared the prize, Nobel Prize in medicine and physiology, and one of them was Dr. Julius, who discovered this TRPV1 receptor, and for this discovery also of its role in pain and temperature regulation. So this is the molecule we work on, and we are delighted to see that it has been lifted up to Nobel Prize heights. This receptor is located on sensory afferents. So you see here a nice picture of a dog. You have the brain and the spinal cord, that's the central nervous system, and then you have nerves either extending or going back into the central nervous system, and that's the peripheral nervous system. And there are different types of nerves there. So small unmyelinated nerve fibers that sense painful signals in the periphery and sense it upwards if it's very painful, they have this TRPV1 receptor. So that's where we want to regulate that by inhibiting it temporarily. And thereby you can regulate pain, as I said before, but also neurogenic inflammation. And then I think in diabetes, it's the inflammation that is very important to regulate. A little bit about our pipeline. We had the blue, the top bar, that's the blue diabetes project that we presented when we listed. We started in 2021, mid-2021. We raised some funds to do manufacture of some more tablets and do these preclinical longer safety studies. And that has been completed. We are actually right now waiting for establishment of some methods to also certify the tablets for phase 2B. It was only certified for phase 2A before. And when that is ready, we are ready to move into the phase 2B study. Also, in the bottom bar, you have this orphan project in orange that I told before. But if we focus on the diabetes, we have had a colleague of us in the company Explico to work on a net product value estimate, and he has also, together with us, described the development steps we need to take to get to market. And the first steps that the preparation for the phase 2B study, and the next step is then the phase 2B, where you see a significant reduction in the risk of failure. So that phase 2B study is really crucial to actually have a better likelihood to get to market, or to have a compound that actually works like we want it to work on glucose, and then hopefully also will have some added on positive effects. We hope to partner after the phase 2B because this is a big development program. Although we don't need to do some big outcome trials anymore in diabetes, it's still big clinical trials in phase 3. So we hope that we can get good phase 2B results so we can candidate for partnership. The next step, phase 2B, is a really classical parallel design, placebo versus 3 different doses of our molecule. And we have, while we have been waiting for these tox results, we have discussed a lot what doses should we pick. The molecule has been in 8 clinical trials before, and in other patients, not diabetics, the maximal tolerable dose has been set to 4 milligrams bi-daily. But what we saw in our diabetic studies was that single dose of 8 milligram was well tolerated, and also in this study of 1 month's duration, 2 bi-daily doses of 4 milligram was very well tolerated. Nothing really serious on the safety side, and only a small effect. So we think we have to both prolong, I mean, that's the whole thing about this phase 2B, do a 3-month study, so longer duration of treatment, but also we want to get up to the highest tolerable dose. And we simply haven't really been able to pick the middle and highest dose that we think are the best levels. The case is that if this phase 2B goes well, the middle dose is the dose you will need for the phase 3. So it's really important that you get to the right dose levels. So we have decided that we start at one site with a clinical investigator in UK. And we will do, and this is his suggestion for a dose escalation study. These are maybe not the final dose levels, but suggesting that we start in a fewer number of patients and follow them and start them on low doses and then until 3 months, and then go up gradually, add on 4 milligram at a time, and to see if how high can we go before it's too much for the diabetic patients. So we hope to move on to that as soon as our tablets are certified, and then we also need to secure some funds, and we're working on that. That's all I can say about that at the moment. But the idea is then to start in a smaller subset at a clinical site where we can define the doses and follow for 3 months, and then add on more sites to get up to these 300 patients in total. So a little bit the summary for our project. The estimated development costs to get to market is approximately 120 million euros estimated, and we estimate that the phase 2b study will cost around 7.5 million euros, and out of that, this dose-finding part, only €500,000. So we will start with that. And for that, you can get to a very conservative estimate, I think, for this project of a value of 1.4 billion euros in total value of the project. We have, of course, the main risk, that is lack of efficacy. So that's why we will add this or do this phase 2b, starting with the dose finding to be sure we have the right doses that are needed to get a good response on effect. We have a good scientific advisory board, and here is the management team, and I say thank you, and you may have some questions.
Perfect. Thanks a lot for your presentation. So I guess we could start then with the ZEN-D0501, and I think you call it ZEN-DIA when you target diabetes, right?
Yeah, that's a project.
Yeah.
Yeah.
And I mean, as you mentioned, of course, the phase 2b trial is very important for you moving forward. And you said that there's still a financing gap to be bridged until that could be started. But what is the status? Is the compound ready for phase 2b if you have the required capital, or is there more sort of preparative work required?
So we are still waiting for last results. So we did manufacture new tablets in 21, actually before we listed, so for the pre-IPO money. And we, on the way last year, realized that we needed to upgrade the analytical methods and have it certified for phase 2b. I didn't know that before, but so we inserted that, and we raised a little bit of funds in November, and for that we could start this work, and that is ongoing, and it's scheduled to end here during the summer if everything goes well. So when that is ready, then we can submit the clinical trial application. And we have a site in the UK with one of our scientific investigators that we are planning this study together with. And, well, we're still discussing a few details, but I think we're more or less ready to move on to that. So, so with money, we can hopefully get going with that. And it's the first part is not so much so in these hard times. I think we are going to maybe do it gradually. Instead of going for the big sum all at once, we may start to fund this dose-finding study, which will also be some kind of show if it's tolerable, and if we can maybe see a sign using continuous glucose monitoring. I think we will do that on these patients and see if we can see a trend. So
interesting, perfect. So I mean, looking at the profile of your molecule, I mean, I guess it would be sort of an early line treatment. Is that correct?
I expect that we must get into early. Post-diagnosis, maybe after metformin. So the trial we did was in what we call metformin failures. It's known that when you have failed on diet and exercise, and I can tell that myself, that's difficult to control also. But if you become diabetic, then you're offered a tablet treatment called metformin. And so after that, you get another tablet, or third and a fourth. So it's in this tablet space we hope to get in. And as add-on to metformin, we can improve insulin at least. So that's first step.
Okay, perfect. And I mean, as you mentioned, metformin is sort of the first-line treatment. It's a generic tablet that has been on the market for a very long time. But I mean, looking at your potential product here, then how would you pair it compared to metformin? Are there any advantages, or what are the big distinguishing factors?
Well, we hope to get a better effect on glucose when they are not well regulated on metformin, obviously. And then, I mean, metformin actually has some good effects on cardiovascular function, so that's why patients are kept on that for even after they deteriorate on their glucose. But we also believe we can have some cardiovascular positive effects. And we also believe that there can be other positive effects related to this reduction of inflammation. Again, maybe I should take a slide next time to show all the things I think we can do something about. We have some indications on positive effects on heart function, but...
Okay. And I mean, I also want to touch upon, I mean, you mentioned it, that you're targeting the TRPV1 with your molecule. And I mean, that is sort of a unique approach for Pila, at least in type 2 diabetes.
Yes.
But can't you please elaborate slightly on the foundation of this idea? I mean, how...
So TRPV1 was discovered by David Julius. I think researchers within the pain space had known for quite a long time that if you used capsaicin, you could modulate the sensation of pain. So in the pain area and inflammation, this molecule, this target has been well known to have a function. And so when I did my PhD, I used capsaicin in a preclinical study in diabetic rats for something completely different. But then I found that their blood glucose was kind of, completely regulated by just a single shot of this capsaicin. And then I got really interested into that, and I tried to follow, and I did a number of experiments, and it turned out that this receptor seemed to also regulate insulin secretion, primarily also insulin sensitivity, and thereby glucose. It also affected food intake, energy expenditure, possibly body weight. There are also, that's not my work, but in the literature there's evidence from preclinical side that it has an effect on gastrointestinal function, even brain function, and so on. So it seems to be, it's a receptor that is on these sensory nerves, and they are all over in the body. So if that comes out of tune, many organs can be out of balance, and so hopefully getting it just down-regulated a little could improve symptoms in many organ systems. So the comorbidities in diabetes, I hope we can address them also. That's why I put my money on this project.
Okay, perfect. So, and I mean, would you describe Pila as a TRPV1 platform company now, or how should it be looked at? Since, I mean, you have at least previously the type 2 diabetes. I mean, it's still your lead project, I guess, but I mean, you have recently been talking a lot about the orphan drug designation, etc.
I think, there have been other companies trying to develop a TRPV1 antagonist, but they have failed due to safety issues. So for some time, there was a lot of interest in developing these compounds, primarily as painkillers, but then a lot of this collapsed because of side effects. Now we have an inhibitor that seems to be safe. And of course, I have my own idea in diabetes, and that's my life project. But we also see that if we have this molecule that is safe, and if it works as it should, as an inhibitor of this very potent and important receptor, then we might have a tool or platform to use in other inflammatory conditions. So I hope, but we have to, crawl before we can walk. Yeah. We're a small company, we don't have a lot of money. So my board has been very focused on first we must kind of, move ahead in diabetes. Now we've added on this orphan that we're also seeking funding for. And we're also working with some preclinical research groups to see if there are other indications we can start to investigate. So yes, we hope to become a platform company. But yeah.
Perfect. So, I mean, we have a few seconds left here, and I want to give you the moment then to be able to present what would you say is the most important milestones and events ahead of Pila, moving into the rest of 2023?
Yes, it's obviously to secure funds for actually both studies. We have seen quite some interest in the orphan project, so we hope, but it's still we are talking to many types of different investors, and soon we will have to sum up and say which way we go. So hopefully we can fund both projects, and then it would be a proof of concept in the orphan, and then this dose finding in diabetes to risk minimize in both projects. And to it's the same molecule, so the project will move secure the other, so to speak. So it's this getting to this next level of characterizing the molecule in pain and showing that we can give higher doses and for longer in diabetes.
Perfect. Thanks a lot, Dorte.
Thank you.